right. Good morning, everybody. My name is Lloyd Tripp. I'm a second-year MGG student. I have the pleasure today to introduce Sean Eddy. Dr. Eddy is the Ellmore C. Patterson Professor and Chair of the Department of Molecular and Cellular Biology at Harvard University and a Howard Hughes Medical Institute investigator. He received his Ph.D. from the University of Colorado Boulder, working in the lab of Dr. Larry Gold. In 1995, Dr. Eddy joined the Department of Genetics at Washington University in St. Louis, where he was the Alvin Goldfarb Distinguished Professor of Computational Biology. While at Washington University, Dr. Eddy helped establish the Computational Biology Program, now CSB. In 2022, Dr. Eddy was elected a Fellow of the International Society for Computational Biology. His lab focuses on computational methods for biological sequence analysis, especially methods to identify evolutionary relationships between protein and RNA sequences. Please give a warm round of applause for Dr. Eddy.
All right, so I'm not doing slides, I'm doing this a cappella. Last night on the plane, I was sitting by a singer who had sung the national anthem a cappella at the Obama White House. And I said, well, I'm giving a talk a cappella tomorrow too. Okay, the first thing I need to tell you is that I tend to get emotional at things like this. So I have to warn you. A few years ago, I was doing a celebration for a friend of mine in the UK. And he was somebody who was a scientist, but he knew everybody, it seemed. He knew novelists and musicians. And so at this thing, there were all kinds of different people. And when it came time to give my talk, I completely lost it. I just completely lost it on stage. And afterwards, my partner Elena was in the coat room, and there was a well-known novelist, Ian McEwan, in the coat room, and he was talking to somebody else, and my wife was eavesdropping, and McEwan was saying, "I've just heard the most remarkable thing. I had no idea that scientists had feelings." And the reason to be all emotional at a thing like this is because it's a special day. It's a special opportunity. There's conversations that you want to have with people who've made a big impact on your life. And there's never the right time to actually do it. You know, it's like when it comes time to do the annual performance reviews for the people in your lab, you actually have to schedule a time, you know, to sit down. The big conversations, they happen like on road trips at night where you're not looking at each other and you're just driving through Oklahoma or something. You can talk about something seriously then. But otherwise, you have to schedule it. And so that's why we're here, is to have this kind of conversation. You want to have this kind of conversation today in public with a bunch of people who can hear you say what you need to say. And so thank you, Ting. Thank you to the department for making today's public conversation possible and bringing us all back together, and for inviting me to be part of it. I've known Gary for almost 40 years. I joined the PhD program in Boulder in fall of 1986 and met Gary then. He had finished his, so, you know, you're a first-year graduate student, you know nothing. Everybody seems like they've been there forever in the department. But Gary had just gotten his PhD in 1981, I guess. He had been a postdoc there at Boulder and had probably gotten his faculty position. I don't even know, I couldn't even find this. Must have been 1984 or something that he became assistant faculty. So he was really new. But to me, it was, you know, one of the faculty, and as I'll tell you, I wanted to work in his lab. Our bios have been weirdly parallel. So not just Boulder, where we both did our PhDs in the same lab, but Gary and I went to the same place as undergraduates at Caltech. He was an undergrad at Caltech where he wanted to be a physicist, but decided to switch to biology in his junior year because he correctly saw that biology was going to be more interesting. And I was a undergrad at Caltech who wanted to be a physicist, but was incapable of doing physics, and switched to biology even faster in my sophomore year. Now, something that's worth knowing about Caltech alums is that the houses, at least in our generation, had very distinctive personalities. And when Caltech alums meet each other, like sort of dogs sniffing each other, the first question is, what house were you in? And you can deduce a lot about a Caltech alum's personality by what house they were in back then. Back then, there were two athletic houses. There was a studious athletic house, very respectable, and there was a not very respectable and not very studious athletic house. And Gary was in one of them, and I was in the other. You can probably guess that Gary was in the studious, respectable house, Page House. So it's not surprising, based on being in the athletic houses at Caltech, that one of my first memories of Gary is actually from playing football. He was a running back at Caltech. Now, Caltech is basically all walk-on. I played varsity soccer at Caltech, so it's not like it's like an intramural program, but he was a good running back, it turns out. And we learned that at an MCDB retreat where there was a game. I'm dimly recalling this, between the first years and must have been the faculty. But I remember Gary's moves, because if you know him, you know that Gary is very deliberate in his motion and in his speech. And this shows up on the football field as well. And so he has this wily move that's very distinctive, where it's sort of a slow slide to the one side, and that makes you think he's going to go slow, and then a very quick move to the other side. And I remember being juked out of my shoes by this move at the retreat. And you learn very quickly not to underestimate Gary. And that's also true about science. So I wanted to rotate in his lab, but so did a whole bunch of people. There was a lottery for getting into his lab as a rotation project, and somebody else won the lottery, and I didn't get to do a rotation with him. I did other rotations, and I ended up in a great lab. I ended up in Larry Gold's lab, where Gary had also done his PhD, and that's another parallel in our sort of academic bios. Larry's here today, and he's going to be giving a talk later this afternoon. The three of us played a lot of basketball together. I think it was every Tuesday, maybe it was every Thursday morning. There was a game at Boulder, and we did a lot of playing. And then Gary had the same move on the basketball court. And I still have that very much pictured in my mind. And I also remember Larry shouting, work the ball inside, which always meant he was going to take a 3-point shot. A surprising thing about Boulder that I've never understood is that Boulder was a nursery for a field that had not even formed yet, computational biology. And it's to this day unclear to me why so many people who are now the pioneers of the field of computational molecular biology had come through Boulder, either through the biology department or through the computer science department. David Hausler had been there. Gene Myers came through Boulder. Gary was, of course, in Boulder. Robin Guttel was doing ribosomal RNA work at Boulder. Tom Schneider was developing information theory with Gary at Boulder. George Hartzell was working with Gary on motif finding, and was one of the people who really was pivotal in teaching me early techniques in computational biology. And Gary was pioneering the use of these things called position-specific weight matrices for recognition of like DNA binding motifs for proteins, and showing that they were way superior to just a simple consensus sequence and stuff like this. And there was just a ton of sequence gazing going on. I can remember sitting in lab meetings where there would be a sequence from somebody, maybe it was a seminar speaker or something, and everybody learned how to sequence gaze. To this day, so it was sort of a game. Larry would recognize, oh, there's an RNA secondary structure there. And Gary would also recognize sequence motifs by eye. And so this was sort of instilled in you that you could get a lot of information from looking at sequences, and it was infectious. And to this day, I can tell you the Shina-Delgarno consensus sequence for bacterial translational initiation, because it was drilled into me by all this sequence gazing. One of the algorithms that is really important for motif identification is something called expectation maximization. Gary was one of the pioneers in introducing EM algorithms for motif identification. And, no slides, I'm not going to try to explain it, but the basic idea is that you start with a random position-specific matrix with random parameters, and then you align it to all your sequences and see what the random thing finds, and take everything that it found and make a new matrix trained on the things that you found, and iterate that until it converges. And magically, this shouldn't work. You're starting with noise, and it converges to a solution. So I was an experimental biologist, but I was working with a bacteriophage where we had the genome sequence. I was dabbling with computational techniques and how I could use them in my work. And I remember sitting in Gary's office, and this was sort of a pivotal moment for me as I was trying to understand this very counterintuitive to me algorithm, this magical thing. How can this possibly converge? Gary very patiently trying to explain it to me as I was sitting there going, no, but no, that can't work. And it does actually. And so now I'm a professor of computational biology, and every year I teach Harvard undergraduates expectation maximization. And I do the same thing for them. It's like, no, it actually works, look. And I use one of Gary's papers as the example as I'm teaching. So I left Boulder in 1992 to do my postdoc. And just before I left, and this is something that I don't even know if I've told Gary, that this is like a pivotal moment in my life. This actually made like a huge difference. As I was leaving Boulder, he gives me this preprint from Anders Krug and David Hausler on what are called profile hidden Markov models. And it was just, we had preprints back then, turns out. It gave me a preprint and said, you should read this. I think you'll understand it, and I think it's going to be important. I said, thank you. I put it in my backpack. I moved. You know, it's just like you Xerox papers back then and never read them. I had this paper, and it was like, one of these days I'm going to read this paper. I arrive in Cambridge, England for my postdoc, and one of my mentors, I was going to do an experimental project, but one of my mentors was Richard Durbin, a computational biologist, and he gives me the same paper, the same preprint. He says, you should read this. It's going to be important. To understand profile hidden Markov models, you had to sort of understand a bunch of stuff. You had to understand probabilities, but Gary and George Hartzell and other people sort of in the community at Boulder had taught me that. You had to understand the expectation maximization algorithm, but I sort of knew that by then. And so I was in a position where I could really understand this and use it in my work. And so from Gary and Richard telling me, you know, you should really read this because we think that this is gonna be something that's important, I started trying to understand this one paper really well that Gary had given me. And that really sort of launched my career in computational biology, was that one paper and that one moment. And I switched from just dabbling in computational biology to doing it full time. So I was in Cambridge, UK. I moved here to St. Louis for my faculty job in '95. It really took leaving Boulder to understand from an outside perspective just how important and influential Gary's work was in founding computational biology and sort of building what we now consider to be its own field. And I think in Boulder, because Gary was a PhD student, a postdoc, then faculty, that, I mean, I love Boulder, but I think Boulder didn't quite recognize what they had. Didn't quite recognize Gary for the work that he deserved to be recognized for. And I think that's typical of academia, and it's something that you sort of need to watch out for, right? There's a reason why we live this semi-nomadic existence of moving from Caltech to Boulder to WashU and all over the place. It's because people sort of assume that if you've been in the same place for a while that somehow you're not fresh or something. I don't know what it is. But when I was here, starting in '95, this place had also become a hotbed for computational genomics for sort of accidental reasons. We had the Genome Project. Bob Waterston was going great guns, collaborating with John Solston in the UK, who was my mentor and my co-mentor in the UK. Warren Gish was here. David States was here. Michael Zucker was here. Phil Green had been here and left, which opened the position that I took. This place had become like the center of the universe, weirdly, in computational, again, in sort of founding comp bio. And I had a feeling that both Susan and Gary were movable from Boulder at that point. And I think that it's fair to say that I had some small part in attracting them here for the second act of their careers. So another parallel for us, both being here in genetics. And I hope that move repaid a little bit of my karmic debt. I hope that it ended up being good for you both. I think it did. Even if St. Louis is maybe a little hotter than Boulder, the weather maybe. I know it was definitely great for WashU. So since that brings us to St. Louis, and I left St. Louis in about 2005 then, I'll leave it now to others at the meeting to pick up from St. Louis. I'm glad, really glad to have had this opportunity to publicly thank Gary for your influence on me at multiple points through my career over the years. Maybe today having the opportunity to pay a little bit more of the karmic debt. Thank you, Gary. Congratulations on an amazing career so far, and best wishes for the next stage of it.